is Hawai'i Nakasone.
Good morning, everyone. My name is Riley Nakasone. I'm a laboratory technician in the Hawai'i DLNR, SEP stands for Snail Extinction Prevention Program, in the captive rearing lab. And today I'm going to be presenting our research titled Feeding Trial Using Auriculella ambusta, a tree snail native to Hawaii, comparison of an artificial diet to that of wild collected vegetation. And so for a brief introduction to kahuli, or Hawaiian land snails, the most recent estimates from our partners at Bishop Museum say that there were just over 750 described species of Hawaiian land snails. Unfortunately, it's been estimated that 60% of this diversity has already been lost to extinction, and within the next 1 to 10 years, without significant conservation action, we're looking at losing another 100 species, representing the near total extirpation of the 11 genera pictured here. And so one tool at SEP that we're using to try and prevent these animals from going extinct is captive propagation. Currently in our rearing facility on Oahu, we have around 40 species representing around 70 distinct populations. And most of these populations are extinct in the wild. And so as these snails continue to disappear from the landscape due to threats including climate change and introduced predators, captive rearing is going to become an increasingly important tool to keep these animals around. And so it's important to try and optimize captive rearing now, because the more efficient we're able to make the process, the more species we're going to be able to save. Right now, arguably one of the biggest limitations on captive rearing is the reliance on native vegetation. Our tree snails, such as this Auriculella lila pictured here, which is actually giving birth, you can see the baby coming out of the body right there. But yeah, these tree snails are fungivores that feed on the fungus and microbial community that grows on the surface of the leaves out in the forest. And so we're having to go out and collect native vegetation to bring back to rear the snails on to provide them their food. And the problem with this is that collecting the veg is time consuming. It requires going out and cutting the veg, and then it restricts captive rearing to areas within geographical proximity to the native veg where someone can go out and collect it and then bring it back to the lab. Another issue with collecting the native vegetation is that it allows an entryway for pathogens into captive populations, and we could see die-offs. So now let's jump to a different group of organisms who aren't taxonomically related to Hawaiian tree snails. However, you could argue that they fulfill a similar ecological niche, and that's the southern Pacific tree snails of the family Partulidae. Similarly to our Auriculella tree snails, Partulids are from small volcanic islands. They're nocturnal, so active at night. They're primarily arboreal, so they live up in the trees, and they're ovoviviparous, which means that they carry an internal egg, and then that egg hatches internally, and then it looks like they're giving live birth, but actually the egg hatched, and then the baby is just emerging. So the group in charge of the conservation of the partulid tree snails is the International Partulid Conservation Program, which is a consortium of zoological facilities that was formed in 1986 to prevent these animals from going extinct. They're facing similar threats to our Hawaiian land snails, such as climate change and introduced predators, most notably the rosy wolf snail. One of the biggest accomplishments of the program is the development of an artificial diet on which the snails can persist exclusively without the need for vegetation, and this has allowed the program to establish 10 species in captive rearing distributed across the globe. And so the objective of our study is to determine if the Parchula diet warrants further study as a supplement and a complete diet replacement for rearing our Hawaiian land snails. You can see our current setup pictured here. We have some cages filled with the native vegetation that the snails feed on. But can we reduce or completely eliminate the need for native vegetation and move to a setup more similar to this Parchula setup that just has a paper towel to maintain moisture, and then the Parchula diet presented on these feeding dishes? And so to assess the Parchula diet, we conducted a 14-week feeding trial in which we collected data on survival and fecundity. For fecundity, we looked at egg laying as well as the hatching rate of those eggs, so that we have a true value of the reproductive output. And then we analyzed our results with a Kruskal-Wallis test to account for our small sample size and non-normally distributed data. So the organism we studied was Auriculella ambusta. I want to stress that we did not test on any of our rare species, and we will not do so until we're confident that the particular diet will not negatively impact them. And so Auriculella ambusta, there's some adults pictured here, and then this species actually lays eggs, and then we incubated those eggs to get the newborns. So Auriculella ambusta was chosen as a surrogate species because they're locally abundant in multiple populations throughout the Waianae Mountains, and they're still in the Auriculella family of true snails. And then compared to our rarer true snails, they have a higher fecundity. So during the 14 weeks of this study, we were able to get nearly 400, just under 400 juveniles. And to get that many Auriculella, for example, would take about 3 to 4 years. So this species allowed us to get quick fecundity data from a non-listed, non-rare species. For our experimental design, we had 3 treatments. The first treatment was a control, just had ohia and a empty feeding dish. This was meant to mimic our current capture rearing setup. And then the second treatment had the ohia, where we included the feeding dish with the partula diet to test what effect the diet would have in addition to the native vegetation. And then our third treatment, we had tea leaf as well as the partula diet. And we decided to include tea leaf because we see snails on tea leaf in the wild. And we thought tea leaf would be a good non-native substrate that did not provide too much biofilm to the snails. Because when you look at tea leaf, it's usually pretty clean. You never see like the sooty fungus or mold that grows on like ohia or llama, for example. So we included tea leaf as a non-native neutral substrate. And we had 3 replicates per treatment and 5 snails for each replicate. So let's look at our results. The only treatment that had deaths was the tea and partula diet treatment, as you can see here in yellow, and we found this decreased survival to be statistically significant. And then looking at the egg laying, we quantified egg laying as egg laying rate given as eggs laid per snails per cleaning cycle. And so we have the control over here in blue, the ohia and partula diet over here in orange, and then the tea and partula diet over here in gray. So as you can see, both treatments with the partula diet had a higher egg laying rate compared to the control, and we found this increased egg laying rate to be statistically significant. And then when looking at the hatching rate, we found that there was no difference in the hatching rate between the control and the ohia and the partula diet. However, when we looked at the treatment with the tea and the partula diet, hatching rate was significantly lower. And so what do these results mean? The first thing that we found was kind of a mistake, and it was a surprise to us that the tea and partula diet decreased survival and fecundity. And so this brought up the question, is tea inherently bad for the snails, or perhaps is it lacking some sort of native biofilm that's present on the ohia that the snails need to thrive? Unfortunately, this study does not completely answer that question. However, I will say that we do have previously unpublished data from another alternative veg study that also suggested that tea was bad for a captive rearing setting. So we're not saying that tea leaf is bad for Hawaiian land snails because we do see them on tea in the wild, but perhaps when we collect it to bring it into the lab, damaging or cutting the leaf causes it to release some sort of chemical that the tree snails don't get along with. And we accomplished our objective of determining that the partula diet does warrant further study as a rearing tool in Hawaiian land snails because egg laying was increased in both of the treatments with the partula diet, and hatching rate was unaffected when fed in conjunction with ohia. In fact, when you compare ohia and partula diet, the treatment with ohia and partula diet to our control, fecundity was increased by 14 times. And so where are we going to take this study next? Next, we want to investigate whether the snails can persist on the diet alone, the partula diet alone, or do they need some sort of vegetative substrate? And does the vegetative substrate need to be non-native? Does it need to be native, or could it be a non-native species, or perhaps even plastic vegetation? And if they do need a vegetative substrate, what quantity of that vegetative substrate is needed? Because once again, the goal of this study is to eventually reduce the amount of native vegetation that we're having to use to raise the snails on. We're also interested in determining if the partula diet is suitable for Hawaiian land snails in different families, such as the amostrids. The amostrids are primarily ground-dwelling snails, and they're detritivores, so they feed on dead and decaying plant matter, mostly nettles. And so right now we're actually conducting a study back at the lab testing the diet in an demonstrative species. And then in the long term, before we test on any of our rarer species, we're going to need to do long-term feeding trials to determine how the diet could possibly affect how the snails grow from juveniles into sexually mature adults. Mahalo for listening to this presentation, and I'd like to give a special thanks to the entire SEP team, as well as the Woodland Park Zoo's Partula Lab for showing us how they raise their Partula snails, and I'd be happy to answer any questions that anyone may have at this time. Yeah.
So they'll lay their eggs without any vegetation to lay them on?
So we did a preliminary test. While we were testing if the snails would even take the Partula diet, we tried taking away native vegetation just for like 2 days. And during those 2-day periods that the snails were presented the Partula diet but not any vegetation, they did not lay any eggs. And then immediately after we added a piece of vegetation, they started laying eggs again. So during that 2-day period, it seemed like they held back their eggs, but we want to do a longer term study in the future to see if they need that vegetation or if they were just like uncomfortable for those 2 days.
Follow-up question. Do you think there's any other kinds of considerations when taking away vegetation, such as behavior? If the goal is to ever release them back into the wild, if they've been raised in captivity without any exposure to that kind of habitat type?
Yeah, for sure. That's definitely a concern. Like, if they're raised on the captive diet, will they be able to transition back to a wild diet? But that's going to be a topic of study further in the future. Right now, we're just trying to focus on whether we can use this diet to reduce the amount of native veg. But yeah, that's definitely a concern we have. Yes?
The native vegetation that you feed them in the lab, where do you get that? Is it?
We collect them from state forest reserves.
Okay, so you get it from the places where
the snails are.
the snails are.
We have a field team. Yeah,
Okay.
they go out to like areas where we have exposures, which is predator-proof fences that you'll hear more from my colleague. She'll talk about them in a little bit. But yeah, while they go out and do work at these exposures, they will collect some veg and then bring it back for our lab to use.
Okay. And so the tea that you have in the Partula diet is just, are you raising it?
Yeah, so that tea was actually not collected from a forest reserve. It was collected from right outside our lab in Pearl City, so pretty low elevation. So that's why we thought that biofilm would be pretty neutral and it wouldn't have the same biofilm as a leaf from up in the native forest.
Mm-hmm.
Yeah. Yes?
You mentioned that by bringing in some like plant material from the forest, that you may be bringing in potentially diseases and things. Is that something that's known to impact the?
So yeah, you'll hear more about this too from my colleague when she presents about captive rearing, but we do see mass mortality events, and it seems that it's something coming in on the vegetation. We're not sure exactly what it is, but we're currently working with Bishop Museum to try and figure out what's causing that. Yes?
At the end of the day, could you use these results for a breeding program for the rare ones that there are no effect? Like, let's say they will never just go to a strict artificial diet, but since eggs, increase 14 times, could that just be a part of the breeding program in the future to bring the numbers up?
We could definitely use the Partula diet as a supplement just to like increase fecundity if we find that they aren't able to persist on the Partula diet exclusively.
Is it a mineral in there or something?
Oh, so yeah, I can go over the ingredients. I had a picture here. So the Partula diet is made up of calcium carbonate and then some koi pellets, which provides a bunch of protein, oat flour, nettle powder. The nettle powder was kind of interesting. We did another veg study, which we did a nutrient composition of nettle, the Hawai'i nettle, so like Urera, Mamaki, Olona, and Olona had a similar calcium content to bone meal. Yeah, so super high. And then it also has a stress supplement, which has a bunch of different minerals and vitamins.